Good morning, everybody. About a year ago, my surgeon, Dr. Michael Brodman, asked me to co-chair today's Women's Health Day of Learning. Gratefully, I accepted being given the opportunity to acknowledge, thank, and support my doctor and the department that is giving me the kind of care we all hope to receive. With 7 hospital campuses and more than 45 ambulatory practices citywide, there's a Mount Sinai physician in walking distance of us all. The Mount Sinai healthcare system cares for everyone in my family, many of whom are sitting here today. I know that they share my feelings of fortune and gratitude to be associated with one of the finest hospitals in the world. Thank you to everyone here for your attendance at today's event. Your generous support will greatly further Mount Sinai's efforts to improve healthcare given to women. It is now my pleasure to introduce our speaker today. Dr. Peter Dottino is the Director of the Division of Gynecologic Oncology, Department of Obstetrics Gynecology, and Reproductive Science at the Icahn School of Medicine. He is also one of the finest physicians in his field. Please welcome Dr. Dottino.
Good morning. Could you turn those backlights down a bit? It's like absolutely blinding. It's really a pleasure to be here, and it's so nice to see so many friendly and familiar faces, and I appreciate the support and everybody. And I think at the end of the day, this is what it's going to take to get us to where we want, and that is not to be having conferences about these kinds of things, and I can be out of a job, which would be really good. I'd like to thank Dr. Brodman and the entire staff of the OB/GYN department for putting this event together and also considering us for a presentation to sort of give you an idea of what some of the risk factors or some of the things you need to know about ovary cancer, and then also to allow us to touch on some of the research that we've been doing, so to give you an idea of how we're moving forward. Every day we make new discoveries, and every day we get closer to putting an end to this problem. So we thank them, Mike, and definitely for his continued support. Also, just a word about the division that we have at GYN Oncology at Mount Sinai. For those that don't know, we have the largest collection of GYN oncologists in one academic institution of anywhere in the country. We have over 14 members that provide just world-class care. So I'm very proud of the division, and the care that we render to patients is just world-class. Lastly, I don't do emails. I don't type well. If anybody has any questions, I will be here afterwards. But if you did want to send me an email, my email's on the Mount Sinai website. Just put a phone number at the end of it, and I will call you because, again, I don't type well. So that being said, let's go ahead and get started. I'd like to cover 2 different broad areas here today for us, and one is that the sort of a basic understanding of ovary cancer to give us all a sort of similar platform to work from, and then once we've covered that, to sort of segue that into how do we answer those questions via the research that we are currently conducting. I'd like, if we could, to sort of, I will definitely save time for questions at the end. So if you could hold your questions to the end, we'll then go through them. So to get started in a broad sense, just to help everybody focus, the 3 most common cancers that are going to affect women in the U.S. today are going to be breast cancer, lung cancer, and colorectal cancer. And I think if you take a global view of the cancers that most commonly affect women, we find that breast, colon, and cervical cancers are the most prevalent cancers and responsible for the most deaths worldwide. And the question always comes up is, how do we screen for these cancers? Or more importantly, how does screening differ from early detection? And when you look at these 3 cancers, breast, colon, and cervix cancers, each one of these cancers has a clearly defined, and I mean a clearly defined precancerous state that allows us to detect, and if we can detect cancer before it starts, that can be interrupted, and you can cure that. And we always use the model of cervical cancer in the sense that if you go for a Pap test, and you get a Pap test every year or every 2 years or every 2 to 3 years, depending upon your particular risk, you don't get cervix cancer. And the reason for that is that you scrape the cells off the outside of the cervix, and if there are any abnormal cells indicating precancerous conditions, those can be eradicated. And also, we know very clearly that the progression from the time that you have an abnormal pap smear to the development of cancer is between 8 to 10 years. So it's a very orderly process. It doesn't just go from an abnormal smear to cancer. That doesn't happen. So that anywhere along those lines, even if there's a false negative rate in a pap test of about 10 to 15%, even if 1 year your pap test was read as negative but was actually positive, if you continue to get it, you will eventually pick up these precancerous conditions. And if we look back, before these pap tests got as good as they are today, 15, 20 years ago, I was doing 75 to 80 operations a year for cervical cancer. Now with these effective screening measures, particularly the good pap test, and again, a screening system that works, I do 2 operations for cervix cancer a year. So screening is really where the ball game comes in when you look at cancers. And if you think also of the model of colon cancer, when you see your doctor, they do a family history, they do a risk assessment, you have a colonoscopy, and what do they look for? Well, they look for polyps. They look for precancerous polyps because, again, people just don't get colon cancer. You get polyps, which gradually develop into cancer, and that process, depending upon your risk factors and the type of polyps you have, can be a very slow process. So some people will have colonoscopies every 1 to 2 years based on their family histories. Other people will have their colonoscopies every 7 to 10 years based on what we understand to be the progression of the disease. But screening has made a huge, huge impact in these diseases. And when you look at cervix and colon cancer screening, these are 2 cancers where screening actually works. And there's no excuse not for doing that. So as a take-home message for that, it is very clear. Also, when you look at breast cancer, with the use of mammograms, what it has done is shifted the detection from advanced disease in breast cancer to very early cancer. And what that has done is allowed for early detection, and early detection, detecting cancer at an earlier stage, that ultimately is going to translate into better chances for cure. So again, what these screening does is it's going to cast a very large net, and we look to capture asymptomatic women with the intent of capturing unsuspected disease. And the screening allows identifications of these precursor lesions, which we can act upon, and that's going to decrease the incidence of cancers, and more importantly also the morbidity and the mortality that goes with these cancers. And although screening can sometimes lead to early detection, by definition, early detection focuses on individuals that are at high risk for a particular disease, or more importantly, those that will have subtle symptoms of the disease that would lead the provider to provide more aggressive care in making a diagnosis before the disease becomes widespread. So by diagnosing cancer also in the early stages, treatment becomes more effective, cure rates are higher, and mortalities are improved. So screening and early detection really is where this is all at. And unfortunately, currently when you look at ovary cancer, neither screening or early detection strategies yet are currently proven to work. So what we're going to focus on are, given that scenario, what are the things that you need to know? What are the things you need to tell your friends to know to increase your chance of early detection? So a few facts. Ovary cancer is the most common gynecologic cancer that affects women, and we're looking at 23,000 cases in the U.S. each year, and that is going to be small compared to the number of breast cancer cases that we see, which is going to be over 240,000 cases of breast cancer a year here in the U.S. When you look at proportionally how many women die, you'll see 14,000 women out of the 23,000 will die versus 40,000 for breast out of the 240,000. So more women are going to die. So this is, in general, a very lethal disease, again, without a way to detect it early. So why does all this occur? And all cancers, when they're diagnosed, survival is going to be highly what we call stage dependent. And that what the stage is going to refer to is where the disease is and how much of the disease you have. It's also a way that allows us as physicians to track how patients do. And for each cancer types, there's always 4 stages. And it's going to very clearly describe the extent of disease, and it allows the provider to individualize the treatment based on the stage and the distribution of the disease. So for ovary cancer, when we look at stage 1, that's going to be disease that's confined to the ovary. When we look at stage 2, that's disease that's spread beyond the ovary to some of the local tissues in the pelvic cavity. Stage 3 and stage 4 disease, the disease has clearly escaped into the abdominal cavity, and in stage 4, it is spread to the lungs or inside the liver. And why this becomes important is that 85% of the ladies that either walk in my door or any of the physicians that treat ovary cancer, 85% when they walk in are going to have stage 3 or 4. Okay? So that paradigm is what we're looking to shift. And the reason we want to do that, and only about probably 8 to 10% of ladies will have disease detected in the first stage, in stage 1. And the reason that becomes so important is that we know that stage 1, our ability to cure these women out at 10 years is greater than 90%. Greater than 90%. And there's actually a subset of stage 1 ovary cancer that is cured with surgery alone. So we know that we can, like with cervix cancer, like with colon cancer, if we can shift from advanced stage to early disease, the impact that we can make is absolutely huge. And I want you to sort of keep that in your minds as we continue to go through. So, again, what do we know about ovary cancer? Again, the cure rates for early cancer are going to be over 90% of those ladies are going to be alive at 10 years. And in a sense, ovary cancer doesn't differ from other types of cancer because early diagnosis is going to translate into good responses. On the other hand, we can compare. If you look, what happens to those ladies who are detected with advanced disease? Well, their survivals are, the national survival rates are about 50% of those ladies are going to be alive at 5 years. Ours at Mount Sinai happen to be at 60% because of some of the new novel treatments we do, but 60% is not acceptable. In order to also to get to that 60% survival rate, patients can have multiple radical surgical procedures, multiple dose-intense chemotherapy regimens that can really alter a person's quality of life while they're being treated. So again, the shift is that we need to come up with a better way to do that. And what makes this so difficult is the fact is that currently we have not identified how ovary cancer begins and how it actually, what the growth rate patterns are. And that's what's made screening and early detection very challenging. The conventional wisdom is that there's 2 current theories about how people actually get ovary cancer. And the leading one that's been around for years is that every month when there's an ovulation and the surface of the ovary breaks and an egg is released into the abdominal cavity, that break in the surface of the ovary has to repair itself. And that something goes wrong in that repair mechanism on the surface of the ovary, which develops into cancer. Now, what we don't understand is what's the time frame on that. We don't know if a person goes no cancer, no cancer, no cancer, and then in a week or a day or a month gets cancer, or is it a slow, insidious process like we see with cervix cancer or colon cancer, where you have a 8 to 10 year lag between your first abnormality in your cells to the development of full-blown cancer. So these are 2 of the things that really stymie us as we begin to try to unravel this mystery. And we know that this mechanism of damage on the surface of the ovary and repair has definitely something to do with development of ovary cancer, because we know things that inhibit or prevent ovulation each month lower a person's risk to get ovary cancer. So what are those 2 common things? Well, women that take birth control pills where you don't have an ovulation each month, there's no damage to the surface of the ovary. That will lower your risk for development of ovary cancer. And also pregnancy, because when you're pregnant, you don't ovulate. So the more pregnancies you have, the more protection you get against developing ovary cancer. Now, with the advent, and we'll touch on this as we move, our ability to do what we call multi-scale genomics, and that is our ability to unravel DNA not only of cancers but of us as individuals. People have begun to look at other sites to where ovary cancer might originate from. And based on some very interesting studies that were initially done at Mount Sinai and subsequently replicated elsewhere, in women that had high-risk fallopian tubes and ovaries removed, and we looked in their fallopian tubes, you could actually find subtle changes that looked like they were the beginning of precancerous changes. And so that led a lot of investigators to develop mice models so that we can investigate that. And then now also with genomics, it allowed very sophisticated genomics and molecular signatures to be done on the distal end of the fallopian tube, which actually sits right on top of the ovary. And when the ovary bursts and an egg is released, what they call the fimbriated end sucks the ovary up. It goes down the tube into the uterus for fertilization. But when we look at the molecular signatures that you find in the distal end of the fallopian tube, they are identical to what we see in ovary cancer. So this is a new theory. It's now been validated in many places, but it's beginning to give us a clue that the ovary cancer may begin to be developed by abnormalities that take place in the fallopian tube. And that's fascinating because if you think about a screening test, how would we be able to get that? Well, if somebody had a DNC or a hysteroscopic procedure, somebody could pass a small tube into that fallopian tube, maybe take a snip of tissue and it could be analyzed. That may be one way to do it. The other way to do it would be take a brush or a cytology. And a paper was just actually published out of Johns Hopkins with a group that we collaborate on, where they would actually place tampons in women, take them out, and then look for gene signatures on the tampon that would fall down from the fallopian tube. And surprisingly, you could actually find one of the oncogenes that is responsible for ovary cancer, a gene called P53, which is in 100% of people that get ovary cancer. You could actually find it in the tampons when you use these molecular techniques that we have. And this is something that could not be done 5 years ago because technologically we were not that advanced. So there's a lot of very, very exciting things that are happening in this field that are just being accelerated, that I believe in our lifetime, my lifetime, that we're going to see some definitive answers to these problems. So let's turn for a second to say, which is one of the charges today, is what is one's risk factors? Are there risk factors that you should know about that could potentially increase or decrease your risk for ovary cancer that you should be aware of, so that when you see your doctor, you can ask him, if he doesn't ask you these questions as he talks to you, that will help you sort of develop a profile to assess your risk and what you can do to protect yourself. So let's just run through these briefly. Age, the risk for developing ovary cancer increases with age, like everything else, from heart disease to diabetes to this to that. And most ovary cancers are going to develop after the menopause, and over half of the cancers are going to be found in women that are over 63 years old. It is quite rare, I will tell you, to see ovary cancer, an ovary cancer diagnosis in women that are under 40, although it is possible in some of these genetically linked syndromes that we will talk about. Obesity. Many studies have looked at the relationship between obesity and various different what we call Western cancers. It would be prostate cancer, colon cancer, and looking at the relationship between dietary fat intake, animal fat intake, and the increased risk for ovary cancer. And if you look at overall, women that are obese have a slightly higher risk for developing ovary cancer. And again, when you look at these risk factors, I want you to think that each person is a pie chart, okay? In our individual pie chart, you're going to have what your risk factors are as we continue to go through them. You're going to have your family history, your personal genetics. You're going to have your environmental changes, your stress changes. And so what we're also trying to figure out is how much does each one of these things weigh in your risk for developing ovary cancer, because we can tell you very straightforwardly that there are women that carry genetic markers for ovary cancer that never get ovary cancer. So it's a very complicated problem. It's a whole nother field of genomics that we deal with called epigenetics, and that has to do with the factors that are around our chromosomes and our genes that actually may have more influence on those than actually the genes themselves. So if we look at the third risk factor, it's going to be reproductive history. And women who have carried their pregnancies to term before the age of 26 have a lower risk than those women who have had their first full-term pregnancies after the age of 35, who have never carried a pregnancy to term. And the overall risk for ovary cancer is going to decrease again with each full-term pregnancy. Oral contraceptive birth controls, as stated earlier, women who use contraceptives lower their risk for developing ovary cancer, and that lower risk begins within 3 to 6 months after starting the pills, and the longer you take it, the more benefit you get, and that benefit is thought to stay with you for a good portion of your life. So it's probably one of the most proactive things that you can do if you fit a profile for risk in terms of prevention or risk reduction. Now, family history of ovarian cancer, breast cancer, and colon cancer is important. And in a percentage of patients, we will actually see that ovary cancer runs in families. So the risk for developing ovary cancer is increased if your mom had it, your sister had it, or your daughter had ovary cancer. And this risk will increase with the number of relatives that have had cancer. And that being said, also the caveat to this is there is probably at least 15 to 20 different cell types of ovary cancer, okay? And there is only one cell type that we see that runs in families. So whenever you talk, or whether we interview people about a family history and we ask about their relatives, we always want to know, is there a pathology report that we can look at? Because before we assign a genetic risk to somebody, we want to know that they have a cell type that runs in their family. And again, path reports, usually from 15, 20 years ago, the way they came information are very important. And that's why these days when we operate on patients, as they walk out the door, here's your pathology report. This is the cell type you had. You put this document in your safe deposit box or your safe, wherever you keep your mortgage documents, because this may be very important 15 or 20 years down the line. So family history is definitely very important. There are also family cancer syndromes, and these will make up 5 to 10% of the ovary cancer that we will see, and these are going to result directly from inherited mutations in specific genes that regulate DNA repair in cells. The hereditary breast ovary syndrome is going to be caused by inherited mutations in specific genes that I am sure everybody is familiar with, called the BRCA1 and the BRCA2 genes. The function of the BRCA genes, in a very simplistic way, because these genes are normal in most people, and the function of these genes is actually to help prevent cancer by making a very specific protein that then circulates in the blood, and that protein actually prevents cancer from developing. So this is actually what we call a tumor suppressor gene, the BRCA genes. Now, if you inherit a mutation in the gene and the gene can't make the protein, then you're at risk to have cell growth and ovary cancer. And your risk will increase to develop both breast and ovary cancer as you age. So, the BRCA genes, the 1 and the 2, are going to be 10 times more common in women of Ashkenazi Jewish descent than in the general population. So again, family history. Where does your family come from? What is my risk? Should I be tested for the BRCA gene? The lifetime risk for developing breast or ovary cancer, if you carry the BRCA mutation, 1 is 35 and 75%, respectively. If you carry the 2, BRCA2, it's 10 and 30%, respectively. The overall general population risk is in the neighborhood of 2%. So it is exceedingly low. The other thing that's important to understand about the BRCA genes, now these are 2 genes that were discovered over 25 years ago, okay, before we had large-scale genomics or anything. And the way these genes were discovered very cleverly was by analyzing families that had breast and ovary cancer that led to the discovery of these genes. Fast forward 25 years, we have what we call the high-hanging fruit, which is the BRCA1 and 2, which is only going to make up, in general, 10 to 20% of those people that get breast or ovary cancer. And so what that tells us is since 25 years ago, we have not yet discovered what genes are going to cause breast or ovary cancer for 80% of the people that get these diseases. It's staggering, but it gives you an idea of the complexity of the problem that we're dealing with. And that, again, one of the things that we specifically do in our laboratory investigations is look for genes that cause breast and ovary cancer in those patients that are known to be BRCA negative, because we want to know what about the rest of the population. If you can be screened for one of those two, people will say, well, what other genes can I be screened for? And as I'll discuss when we get to that part, we're beginning to weasel down on that so that we can come up with a better answer to that. So given this background, the questions to us as providers are, how can we effectively screen the population at large for ovary cancer? And more importantly, what tools can we employ to diagnose ovary cancer at an early stage when the symptoms first present? And finally, what would be an ideal time to institute preventive therapy to prevent ovary cancer from developing? So the how, what, and the when have become so difficult to answer, but in this new era of technology, we are beginning to see progress, I can tell you for sure. So when we look at the development of a screening test, and again, remember what a screening test means. It means that we can find something that a person has, an abnormality that is not cancer, but that if you leave it be, will turn into cancer, in the analogy of cervix cancer or colon cancer. And again, we don't know what that state is for ovary cancer. We don't know what that defined precancerous condition is. But once we do, and we can identify that, and that may come from our understanding of the genomics of the fallopian tube, as I said, and looking for these molecular signatures, that may very well be our inroads into that. But as far as a screening test goes right now, that is still in the works. So what about early detection? If you can't screen for it, is it possible for you to find my cancer early in stage 1, where my chance of being cured is over 90%? And how can we aggressively find this? Because rather than serendipitously, because if you look at almost all the people that are diagnosed with stage 1 ovary cancer today, the 8 to 10%, they're almost all picked up by mistake. And what does that mean? Well, somebody goes for a gallbladder surgery and they happen to look at your ovary and they say, oh, it's not normal. And that starts a bunch of investigations. Or somebody has a backache and they take a x-ray of your back and they see an abnormality of your ovary. So these are cancers that are just not picked up. And I'm going to show you that there's actually a way that these can be. So also about 25 years ago, or 20 years ago now, there was a blood test that was developed in Boston, and it is called CA125. And the reason it's 125, they had to test 124 things before they found one that worked, and 125 was the most common protein in the blood of women with ovary cancer. And that test, we would have hoped that we would have a CA150 that would finally answer the question, but they've not gone behind 125. And initially when the test was developed, we'd hoped that this would be a screening tool, that you could take a blood test, pick up an abnormality, and that would lead to the detection. Over the years, we found that this is not how this blood test works. And the reason is that, first and foremost, is that it turns out that only 80% of people with advanced or early stage ovary cancer have the CA125 in their blood. So 20% of the people that are going to present with ovary cancer, either stage 3 or 4, the CA125 test is going to be completely normal. So that's not going to work as a screening test. Second is that the CA125 blood test is going to be elevated by many conditions that are non-cancerous. And those are going to be, number one, fibroid tumors, benign fibroid tumors can make CA-125 levels very elevated. Endometriosis, a very common ailment amongst young ladies, can make a CA-125 very, very elevated. Also, if you take a CA-125 randomly during different times of a normal menstrual cycle, it can appear very high and very low. And so you will have well-meaning doctors that will take the blood test as a screening tool, and then you're presented with a level that is clearly abnormal. And then the question is, what do I do with that? And the first answer is, well, you shouldn't have had the test. But once you've had the test, it's very hard to dissuade the whole set of information that sort of needs to be sort of clarified as time goes on. So therefore, as a screening test, the CA-125 is exceedingly limited because of the false positives. And also, again, a normal test doesn't mean that you don't have ovary cancer. So it does have it. The only one indication, or the current use of a CA-125 blood test, is that when we operate on somebody for ovary cancer, if while we're in the operating room we discover they have ovary cancer and we take a blood sample and the elevation of that test is correlated with their cancer, that test then becomes the most reliable way to follow their response to treatment and also their risk for their disease to come back. So that is the one currently FDA-approved use of the test, and in that setting, the test is very accurate. But as I show you at the end, we've actually come up with a better way to do it than CA-125 to look at that. The next question everybody's probably saying, well, is he going to say something about sonograms? Should I have a sonogram as a screening tool? And would that help me detect ovary cancer early? And again, the use of a sonogram for screening early detection, again, is limited by the fact that sonograms are not going to pick up microscopic precancerous changes in the ovary. Sonogram studies are going to show that if you see something that's abnormal, that you are early in the detection phase of that. And the other biggest problem with routine use of sonography is, again, not knowing what the time frame it takes to develop ovary cancer. You know, again, does it slowly, insidiously develop, or is it very quick? What would be the proper time to do a sonogram? Should I have a sonogram every 3 months? Would that help me detect something? Should it be 6 months? Should it be done at 1 year? And in fact, when you look at all the worldwide studies that have been done, and a lot of these population-based studies have been done actually in Europe, where healthcare is very centralized, and they can screen 20,000, 30,000 women using sonography to see if it will pick up ovary cancer early and shift that detection from stage 3 to stage 1. It actually doesn't work. What it ends up doing is generating a fair amount of what we describe as unnecessary surgery where you don't find cancer. And no matter how good you think you are doing surgery, if you do enough of it, the complication rate has to slip in. So as a screening test, sonography is not there. The most important thing is what I'm going to tell you now, and this is something that I think that you want to remember very clearly when you leave here. And that is ovary cancer for years, for every textbooks, when I was in school and so forth. It's always been described as the, quote, silent killer, as a disease with no symptoms, and the next thing to know that you were dead. Now, surprisingly, in 2004, okay, and that's 10 years ago, there was a seminal publication in the Journal of American Medical Association, JAMA, and the publication was so important that it made also the front page of the New York Times. And what these investigators did is they went and they spoke to ladies who had stage 1, 2, 3, and 4 ovary cancer, and they began to query them as to what symptoms they had retrospectively, okay? And the most interesting thing that they came up was that they all had symptoms, okay? And the symptoms would go unrecognized, or patients didn't quite make sense of them for 3 months, and then physicians would ignore the symptoms for another 3 months, okay? And so the take-home message, and the interesting thing is you say, well, maybe they didn't have so many symptoms if they had stage 1 disease versus stage 3 disease, and it turns out not to be true. And if you look at it, in contrast to what is previously published in all the textbooks that says 89% of women with with stage 1 disease don't have symptoms. Well, in this study, 89% of women with stage 1 cancer actually have symptoms before. And 97% of women with stage 3 and stage 4 disease have symptoms beforehand that, again, were either ignored or just recorded and thought, well, I'm getting older, I'm aging. And then when they go to the doctor, it's, well, okay, maybe you're menopausal, let's do this, let's do that. And the bottom line is, if you don't think about ovary cancer and you don't ask about it, you won't find it. And so what are these symptoms? If you looked in this article, they could be anything. 77% of the patients had diffuse abdominal cramping that lasted more than 2 weeks. Gastrointestinal, either constipation or diarrhea, 70%. Pelvic pain, 58%. Urinary symptoms, which are more common when we age, especially in women without estrogen, the bladder begins to shrink. We don't have as much elasticity in our bladder. Urinary frequency becomes far more common. And pelvic discomfort for about 26%. So everybody in the room has had these. So how do you know that these symptoms are important? Well, the take-home message, if you have symptoms that escalate and don't go away for 2 weeks, you need to see your doctor. Because again, symptoms are present in early stage and in late stage. And again, as we said, early detection in stage 1 leads to increase in survival. So understanding and recognizing the importance of these symptoms allows for early detection and survival in patients without a doubt. And I will tell you that my mentor, Dr. Carmel Cohen, came up with a very clever line, and I think it sort of sums all of this up, and I'm going to mention it a few times today. And he says, if you have this constellation of symptoms and you go to your doctor and you don't feel that you're making a connection or getting an answer, you say to him, prove to me that I don't have ovary cancer. These are the times we're sending somebody for another test, a sonogram, maybe to see a gynecologic oncologist. Did you have a rectovaginal examination? Did they examine the rectum and the vagina at the same time in order to feel the subtle changes that could tell somebody that you might have a problem? But you don't leave unless you ask that question. And don't say, well, you're getting a little older, so that's why you're getting a little bit of spread here. You have to open up your clothes a little bit. Sometimes that's not normal. And so the public awareness, and it's still, again, this is in 2004 that this information came out, and it's still not commonly disseminated. So I think that really, really becomes important. And remember that. Say, look, tell me, how do you know I don't have ovary cancer? I've got 3 of these symptoms, and I've had them for 2 weeks, and I don't feel well. I think that's important. So let's look at prevention for a second. If we look at prevention for ovary cancer, there are no vaccines that are currently available for prevention. We know that tubal ligation or oral contraceptives will lower the risk. And there is recent evidence, again, as we spoke about, that if ovary cancer does indeed arise in the distal end of the fallopian tube, the fallopian tubes can be removed as a preventative strategy. And in fact, in high-risk women in France, there is currently a protocol that's being done. And these are ladies at a high risk in their early 30s that carry BRCA1 and BRCA2, where removal of the ovaries at a very early age leads to premature menopause and various other symptoms that go with that. These women have signed on to institutional review board consent forms and have had their fallopian tubes removed and are part of an ongoing study to see what happens to them over time. So I think that that's something that we are going to see more and more of as we go forward, as we move forward. So again, routine screening methods are not endorsed by the National Cancer Institute. So it's up to you to say, what symptoms do I have? And tell my doctor, your doctor, that I don't have cancer. So one of the things that we're currently doing at Mount Sinai is we have what we call a high-risk assessment program in the Division of GYN Oncology. It's run by Dr. David Fishman. This protocol will be up and running in probably about 6 or 8 months. And what we are looking to do is there are certain dyes that can be injected that are tied to small nanoparticles that will allow us to visualize abnormal changes in the vascular patterns, either in the fallopian tubes in the ovaries, that may be associated with development of ovary cancer, that can then, you would come in, you would have an injection of the dye, you'd wait about an hour, and the dye is safe. It's currently used for all cardiac procedures. And then you would have high-resolution ultrasonography looking for subtle changes in the ovary of the fallopian tube that would give a clue to what might be going on as a screening test. And those that come out and, that have access to the departmental newsletter that comes out, as that opens, we would love to have you enroll in that. For the last part of this, I don't know where we are for. What I'd like to tell you is some of the interesting stuff that we've been doing in our laboratory for the last 7 to 8 years in our effort to sort of help unroll this problem. In your book, you've seen I partnered with a Dr. John Martinetti, who is a geneticist, an oncologic scientist, and is a gene discovery guy. And it took me 2 years to convince him that this was a very important thing to do. But with Dr. Brodman's help, we've been able to convince him, and now 100% of this laboratory, of which we have 15 people, including postdocs, PhDs, are working specifically on this problem. And what we've been doing for the last 6 to 7 years is every time we operate on a patient with ovary cancer, we have them sign a consent to enroll in our studies, and we collect their blood, and we collect their tumor tissue, and what we do with those is the following. The tumor tissue we actually take to the laboratory, and we develop a cell line, so we immortalize that person's cell line, so that as we discover new drugs that we want to test, we can unfreeze their cell line, do testing on that patient's cell line. Also with the cell line, it allows us to do very much testing on novel drug development. And we've been working with a variety of different companies that develop new drugs, and they want a way to test the drugs. And so they'll bring us the drugs, and we will test them in these various different models, because we will have various different cell lines. We currently have over 300 ovary cancer cell lines, which is the largest collection anywhere in the world of ovary cancer cell lines. And so what we're looking at also is, why do certain patients develop resistance to chemotherapy? We know currently that when ovary cancer grows, it actually goes, the growth patterns are through 5 distinct different cellular pathways. And these pathways are now clearly identified, but what we don't understand is why one pathway is turned on, one is turned off, because the development of drugs has gotten so sophisticated that we now have drugs that will work on this pathway or this pathway. But what we found in ovary cancer is that the cells are very clever, so that if you give a pathway inhibitor on this pathway, it will turn this off, but it's like backing up. If you see a roadblock on a street, you go down, it's blocked, you back up, and the cancer goes right down the 2nd, 3rd, or 4th pathway as quick as within a week or two. That's how clever the cells are. So with the use of these cell models, we're beginning to develop strategies so that you can figure out a way to trick the cells where if you go down one and you're blocked, by the time you think about going 2, 3, or 4, we've already blocked those. So it's very, very fascinating to be able to work with these cell lines. And then the other thing that we've been doing is that, again, on all of these tumor specimens, we've been doing in-depth genomics sequencing, where we do a genetic sequencing of the entire tumor DNA. So we look at all the genes that we do with that. And the arrival, I guess about 8 years ago, of Eric Schatt in the Department of Genomics, and again, through Dr. Brodman's help in convincing them that we were worthy of working with these guys, it has accelerated our work just unbelievably. To give you an idea, 8 years ago, it would cost us $22,000 of grant money to do a single specimen, tumor specimen for DNA. You fast forward today, it's around $1,000. So our ability in the last couple of years to do sequencing and look for genes has just gone off the charts, number one. And number two, Dr. Schatt has assigned 10 people to work with our already 15 people in the lab that are not doctors. These people are PhDs in applied mathematics, computer science, and these are people that have expertise in developing computerized models that allow us to analyze vast amounts of information and try and make sense of it. And without their help, and these people are dedicated, you know, they've been to the OR, all of them, they've seen the nature of the problem, and the work that they do has been invaluable. And what we've been able to do so far is take this vast millions of gene pools that we have, and we've begun now to drop it down, and we have clusters of genes now where we have some clusters that are common to epithelial ovary cancer of, say, 35 genes. So we've gone from millions and millions of genes, not to a single gene, but we're down to about 35. And so what our work now becomes is, out of that 35 genes that we find, we need to find those genes that are what we call driver mutations. Those are the genes that actually drive the cancer, or which ones are passenger mutations, which ones are just along for the ride. And with the help of these bioinformatics people, again, this is a problem that we'll solve. And I'll just give you an anecdote of how that worked. Is that Dr. Shatter assigned this, he said, oh, you know, we got this new person, and I think he's going to be great for you guys, and he's got to come work with you. And so, okay, so it takes about 3 weeks to get a meeting with this guy. The guy comes into the meeting, shorts on, under 30, T-shirt, Mickey Mouse backpack, sits down at the table, and we're like, oh, man, this can't be, right? So he opens up the backpack, and he takes out 2 laptops, right? Opens them up, and they're going, wait a minute, 2 laptops. I've never seen that before. Okay, and so he then begins to ask myself, John, and the rest of the team members the most pointed, in-depth questions about the research that we are doing based on the papers of ours that he has read and based upon his reading of ovary cancer. And one of his previous jobs was he had worked at MetLife, and he'd also developed multiple different software companies for analyzing huge amounts of information and making sense of them. And he felt that with the amount and the chaos that we see in the gene patterns of ovary cancer, this was an ideal thing for him to look at. And so this is another overlayer person. It also turns out, so we're there for about an hour, good meeting, doesn't say anything, closes the laptops, puts them away, says it's time for lunch. And we're looking at this guy, you know, we don't eat lunch here. You know, this is, you know, we work. So he leaves and we look him up. So this guy was the number 35 hire at Facebook, okay? Subsequently left Facebook, formed 2 of these bioinformatics companies, and now he's working at Sinai because he's interested in solving these kind of problems. So these are the kind of resources really that the department and the institution are really bringing to bear on this problem that I think are really going to make a big impact. And one last thing that I'm going to tell you about, also in a similar vein, is that based on this sequencing work that we've been able to do, we have begun to find very specific gene signatures for ovary cancer patients. We will find things that are called gene fusions, where 2 genes are actually welded together. We'll find specific deletions or point mutations. And 2 summers ago, we had a high school student from the Bronx High School of Science, who we get these students every summer. And we'd had a project that none of us could figure out, but we figured there was something there, but we couldn't figure it out. So everybody says, oh, let's give it to Leo. He's the summer student. Okay. So after about 4 weeks, he comes into John's office, and he's got this piece of paper. And he says, you know, I think, you know, and so we're both looking at him, and we look at him, and it's like instant recognition, like, holy shit, this guy's figured, he's seen something that we couldn't see in this mass of gene sequencing. And so he was the one that developed this thing called the gene fusion. Now, the importance of that gene fusion, once we were able to identify that that's what actually caused this patient's cancer, it allowed us to develop what we call a primer, and that's something that would actually match up with the gene. And once we had the primer, we could then take the patient's blood and actually find pieces of the patient's cancer DNA in her blood. And we reported that 12 months ago in a journal called Neoplasia, and it was the first time that was described in ovary cancer. And so what that has led to now is really to develop a personalized medicine approach, because now we can identify, based on the original sequencing, what gene you have and make a primer to it, and that actually becomes far more sensitive to follow your progress than a CA125. Because what we found, and the purpose of having, collected your blood samples for 7 years in our biobank, is that we were able to go, when we found this gene fusion, unfreeze this patient's blood from 7 years ago and actually find that mutation in her blood when her CA125 said she had been cured. And the mutation has never left for 7 years. So it's also changing, because the question is, if we can find enough of these, we can find enough primers that could potentially be an introduction into a screening tool. So I think I'm going to stop there, and I'll take any questions. Yeah.
Is there anything that one can do to live preventatively?
Yeah, I think the weight reduction is shown to improve everything. There's no question. It lowers your risk for all types of cancer. And that means animal fat, animal protein. And also now we're understanding more and more about the deleterious effect of sugar. Okay, and sugar may actually turn out to be, from a cancer point of view, based on something that was discovered at the National Cancer Institute, again, in 1930, called the Warburg effect, that tumor cells and cancers grow in the presence of sugar. So sugar may actually turn out to be worse than animal fat. So as you look to make those reductions, and again, it's body awareness. If things don't seem right, we all live in our bodies. That's the most proactive things that you can do. And again, look at your family history. Are you a candidate for testing for BRCA genes? Do you have enough of a family history that would warrant that? Yeah.
If my daughter, who does not carry the gene from BRCA1 and 2, and is on long-term birth control, how long a period is recommended? She's 30, we're going to 32 now. She's been on probably for 10 years.
You mean how long to stay on the pills? Forever, until she's ready to have children, because the pills actually have a better safety profile than pregnancy, ultimately. And if it gives you that benefit, there'd be no reason, particularly with these new low-dose pills that are around, it almost makes it no sense not to.
I understand that they don't recommend you being on the birth control, although after a certain age, after 40, do you feel the same way? Does the benefit kind of outweigh?
No, I don't. And in fact, there are some ladies that will suffer from, say, perimenopause, and low-dose birth control pills are a very easy way to sort of affect the transition into a natural menopause, and it's perfectly safe. Yep.
I could be incorrect here, but if you are BRCA1 or 2 positive, it appears that the prescription for chemo, the drugs that are used, seem to work better than if you have an environmental ovarian cancer. Is that correct? And if so, why?
It's a great question. The question is, if you happen to get cancer, say ovary cancer, and you happen to carry a BRCA1 or 2, you do survive better. And the initial studies are very interesting, and it circulates around probably not the BRCA1 and 2 genes, but there's a gene on the chromosome that comes right before that we were sort of co-discoverers on, called the CHEK2 gene. And that CHEK2 may actually be the regulator for that may have something to do with that. So it's an unraveling story. We still don't know the answer, but there is no question that people that do carry the BRCA1 and 2 genes have better responses, and survival is increased. Now, can that survival benefit, this is what's not known yet, can that be overcome by more aggressive original surgery, where you say you've seen a gynecologic oncologist, you've had all your disease removed at your original surgery, that may be even a bigger co-founder. So there's a lot of other things that may be involved in that are currently being looked at, but it's an excellent question. Yeah.
Do you foresee gene therapy being used to fight? Do you foresee gene therapy being used
the question is, do we foresee gene therapy, and what does that mean? But I think that what you're probably going to see is a couple of different things, and that will be, first and foremost, where you're going to go and they'll take your blood sample and they'll give you a credit card that looks like it, and that credit card, you go to your doctor, you put it in, and it's going to tell you what you're at risk for, what you're not at risk for, and then based on those things, there'll be certain strategies that will algorithms that will direct you. Some of the gene therapy protocols over the years where they've done that, they are very, very complicated. There's been a number of deaths in those kinds of protocols where you try and insert genes into somebody to correct the problem. One of the other ways to do it, if you look at the model that was actually developed at Mount Sinai by Bob Desnick, who was, the father of identifying these inherited metabolic diseases that are common amongst Jewish patients. And once you find the gene and you understand the pathways at which it works, you can then develop drugs that fix those problems and that are very easy to take. And when we first got this, the what's called a PARP, a PARP molecule that is designed to fix some of the deficits in the BRCA gene. And this is a drug now that's in clinical trials, both in Europe. We just opened the trial at Mount Sinai this month, and these are designed to help the repair mechanism that is wrong in the mutated gene fix itself. The disconnect is that the early results of these trials are not matching, but I think ultimately you're going to find that there'll be various different strategies to correct defects. Okay, thank you.